Hey guys, we're here for chapter 12, which is all about nervous tissue. So, just in the way that we looked at the integumentary system, where we looked at the tissue first, and the muscle system, where we looked at muscle fibers first, we are going to be looking at nervous tissue here for chapter 12 before we dive more into the different organs in the upcoming chapters. So, let's get started. Here are our objectives. This is going to also bleed into some of the other chapters we talk about, but essentially we want to know how we organize the nervous system. The major organs, of course, we'll get more into those in the upcoming chapters as well. What the nervous cells in nervous tissue do, and of course, how they signal or that signal is carried through the nerves. So, the nervous system as a whole is really there as a communication and control system. It is taking stimuli from the environment, internally or externally, moving it to the control center. The control center is acting. How is it doing this? Well, first, it needs to collect the information, again, detect the stimuli that is going to send those signals to your spinal cord and your brain. Then it has to process and evaluate that information. So it has to determine what needs to happen. And then finally, there's going to be this initiation to respond. So the brain and the spinal cord will send this motor output via the nerves to effectors. So the things that will actually do something, which are your muscles or your glands. When we talk about the nervous system, we break it structurally into two parts. We will have the central nervous system that we call the CSN, and that is going to consist of just the brain and spinal cord. And I try to remember that the central nervous system is the brain and the spinal cord because that's sort of like the center of your body. If that helps, go with it. And then there is the peripheral nervous system, which is going to be your nerves and the ganglia. So all of the like little reaching out aspects of the nerves, the ganglia. That is your peripheral nervous system. And I like to remember that as it being on the periphery, like in my arms and legs. So this is how we organize our nervous system when we talk about its structure. We have the CSN and the PNS. Then there is this functional organization as well, where we have sensory and motor neurons. So your sensory nervous system is also called your afferent nervous system. So that is where we receive our sensory information from receptors and send it to the central nervous system. Within that sensory nervous system, you have what we call somatic sensory system, which is going to detect stimuli that we can consciously perceive. And then there is the visceral sensory system, which is stuff that we don't notice. It's going to be our organ function, basically. So think viscera, meaning organ. So this is going to be signals from organs. Then you have the motor nervous system, which is called the efferent nervous system. And that is going to be what initiates a motor output. And then it'll be transmits to the central nervous system, or from that central nervous system to the effectors. So within our motor nervous system, we break it down to be the somatic motor system, which is voluntary signals to skeletal muscles. So if you want to run or pick up a pen, that's the somatic nervous system. Then the autonomic motor system, sometimes called the visceral motor system, that's involuntary things like to direct your heart or the smooth muscles in your intestines or to glance. That autonomic also has a breakdown to what we call the sympathetic or the parasympathetic, and we'll get into those as well. So here's really what we want to know when we're classifying this nervous system as a functional being. You've got your sensory, which is the input, the motor nervous system, which is the output. Within the sensory nervous system, you have the somatic sensory system, where it's things that you perceive, whether it's, light that is making an image from the room around you, or it's things that touch your skin, it's sound that you're hearing. That's somatic. Then there's the visceral sensory, where you are not perceiving it, but it's dilating your blood vessels or commanding your heart or your small intestine to move the food along. Um, so that is all sensory nervous system. The motor nervous system, that's what's going to be doing the response or the change. So it's coming from that central nervous system to the effectors. That is going to be things that are somatic, that's that you control voluntarily, like moving your arms and legs, or things that you don't consciously control, like your heart beating or glands. Okay, so now that we've got our breakdown between the central nervous system, the peripheral nervous system, or whether it is functioning somatic or visceral, if it's sensory or the motor, let's look at the nerves themselves. So, a nerve is just a bundle of axons in our peripheral nervous system. Nerves actually are wrapped in connective tissue. We have epineurium, which is going to be over the whole nerve. It's a layer of dense, irregular connective tissue. We have perineurium, which wraps up a fascicle. A fascicle is just a area of a bunch of axons in a nerve. That is also going to be dense, irregular connective tissue. And then we have endoneurium, which is going to wrap an individual axon. And this is a very delicate layer of areolar connective tissue, and it is going to separate and electrically insulate the axons, because we will be seeing shortly that we are moving a membrane potential down our neurons. So this endoneurium is protecting and insulating that axon. Nerves are vascularized. There are blood vessels that will branch through that epi and perineurium, and then they become capillaries, which is going to allow for exchange between those axons and the blood. So we are having vascularized tissue when we talk about our nervous tissue. And here is just sort of a diagram to show you that epineurium all the way around the nerve here. Then you've got your perineuriums, connecting these axons here. And then that endoneurium is around just one of these axons, insulating and protecting it. Okay, how do we classify our nerves? First, we have cranial nerves. Cranial nerves extend directly from the brain. Then we have spinal nerves, which are extending from the spinal cord. If we want to classify them by what they do, their function, we've got sensory nerves. Sensory nerves contain sensory neurons. They are sending signals to the central nervous system. Motor nerves contain motor neurons that are sending signals that come from the central nervous system. And then mixed nerves have both sensory and motor neurons. And most of the nerves that we are going to name and talk about and look at will actually be mixed nerves in that they are able to both send and receive those signals from the central nervous system. What makes them special is that they'll have an individual axon that will only transmit to, or an axon that will only transmit from. We're not mixing the functions at the axons. So it'll only transmit one kind of information. When we talk about a ganglia or ganglion, that's just a cluster of neuron cells, and it is going to be in our peripheral nervous system. So, what makes a neuron so special? Just like we talked about with our nerve or our muscle fibers, our nerves and neurons have some special characteristics. They are excitable. They respond to stimuli. A stimuli will cause a change in the cell's membrane potential. They are conductive. They have this ability to propagate or send an electrical signal. They have what we call voltage gated channels on the membrane that will open one right after the other in order to send that electric signal. They are able to secrete or they release neurotransmitters in response to that conductive activity. And that secretion of those neurotransmitters, those are messengers that get released from a vesicle, and they will go and do a job to influence another cell. They have extreme longevity in that the cell can live throughout the person's entire lifetime. They are amitotic, which means after fetal development, mitotic activity is lost to most neurons. So you've probably heard that, like, you drink or do drugs and that kills your blood, your neurons in your brain, and then they don't grow back. That's mostly true, that those cells are amitotic in your nervous system, but we will see that there are some that have the property of regeneration, but that's coming shortly. So what makes a neuron a neuron? Like, what are its parts? Well, it has a cell body that we call a soma. It has a plasma membrane that is going to enclose the cytoplasm, and we call that plasma membrane a pericheiron. A neuron will have a nucleus. It's able to initiate graded potentials, or basically receive signaling from dendrites in order to create potentials to the axon. And we'll see what all of that means. It sounds very confusing now, but basically, like we said before, it's able to conduct and send a signal. The soma also has what we call a chromatophilic substance, also called Nissl bodies, made of ribosomes. So our neuron has ribosomes. Then there are the dendrites. Dendrites are these little short, non-myelinated, so they don't have this outside coating, little fingers or processes that branch off of the cell body. Dendrites are where we receive input and transfer it to the cell body. Then there is the axon. The axon is got these long processes or long fingers that come from the cell body. The axon makes contact with other neurons, muscle cells, or glands. The axon is going to attach at the cell body in a place called the axon hillock. It's sort of this little triangle region on the soma. The cytoplasm is called axoplasm, and the membrane is called the axolemma. Those little splits and branches that happen at the axon are called axon collaterals. And when it ends, it has axon terminals or telodendria. The very tip of those axon terminals are called the synaptic knobs or terminal boutons. The synaptic knob is where you have synaptic vesicles that have the neurotransmitters. And so, really, the main job of that axon is to act in a response to those action potentials and then release the neurotransmitters at the synaptic knobs. This last little piece we have to look at here is the cytoskeleton. So we know that we've got cytoplasm. So we've got these microfilaments and intermediate filaments and microtubules inside of that cytoplasm, whether it's in the cell body or the axon. And they're special because we call them neurofilaments because they're in this nervous tissue. And really, they're going to group together to make neurofibrils, which is going to provide strength to the neuron. So here's our typical neuron. So we've got several here like in a line, but let's just kind of look at this one. So the first thing to know is that these little spider webbed off areas here, those are your dendrites, okay? Dendrites. And here's your cell body where you've got your nucleus and you've got all of your ribosomes. And then it's sort of this little triangle area here where we now have this axon area where we lead to these axon terminals. If we zoom closely into those axon terminals, that's where you will see vesicles filled with neurotransmitters, and that is what is released as this conductivity, this electrical pulse is passed down the neuron. And this figure comes from your book, so if you want to look at it more closely, you can pull it up. It's figure 12.3. All right, so how do we transport or move this potential through these nerves? This gets a little bit complicated, but we'll try to break it down. And there are videos for you as well on the D2L in the extra resources if you need more about this. So axons, they move material to and from the cell body. We have what we call anterograde transport. That's from the cell body. I like to think A in anterograde and A in away. So from the cell body. And that is going to move like newly synthesized things away from the cell body to the synaptic knobs. Then there is retrograde transport, where it's moving it to the cell body. So that is going to move materials in the axons that need to be broken down and recycled in the soma. And this can only happen, or I guess this does happen, by fast transport or slow transport. Fast transport is fast, okay? It's happening at about 400 millimeters a day. Like we can actually measure how far a substance is moving. It requires microtubules. There is going to be motor proteins that use ATP. And with this fast transport, either away, that anterograde, or to the cell body is possible. Okay. And if it is away, anterograde, then we're going to move things like vesicles, organelles, glycoproteins. If we're going to, we can also have any sort of used up vesicles and anything that might be harmful. Slow transport is much slower, like max 3 millimeters per day. And this is going to be for moving things from the cell body toward the synaptic knob, so that antero away type of movement. And that'll be things like enzyme, cytoskeleton. If we need more of the axoplasm, which is the cytoplasm for the nerve, that's going to be slow transport. Okay. So, with that, we need to classify our nerves not just by how they move things, but also structurally too. In that, we're looking at how many processes or little fingers are coming off of the soma. So, if we've got multipolar neurons, that's a lot of dendrites that are coming off in one axon. This is the most common type. Then there are bipolar neurons where you've got one dendrite and one axon. That is not incredibly common. Really, it's in your eye that you'll see that. Then unipolar neurons, or sometimes we call them pseudo-unipolar, that is that you've got one process that extends from the cell body, which will then split. And you've got a peripheral process that is going to go into several different receptive dendrites, or a central process that can lead to synaptic knobs in the central nervous system. And then you've got anaxonic neurons, which have dendrites, but they don't have axons. So they receive, but they don't send out neurotransmitter. Functionally, this is according to the direction that they will send their action potential. You've got sensory neurons, or the afferent neurons, which conduct input from the somatic and visceral receptors to the central nervous system. Most of them, and for everything that we want to talk about, we'll just say that they are unipolar. And if you go back, remember, unipolar, one process from the cell body can split into peripheral and central. Okay. Motor neurons, these are the efferent neurons. They are going to take a signal from the central nervous system, send it to somatic and visceral effectors. Those are multipolar. And then we've got interneurons. They're also called association neurons. They receive, process, and integrate information from the other neurons. So from the sensory neurons, from the motor neurons, they're getting information and they communicate between the two. And these are mostly in the central nervous system. And they are multipolar. So here's what we mean with that. We've got this image here, like that's your spinal cord, central nervous system. You then feel something on your skin. Okay, that is sent here, and that is going to the central nervous system. Okay, that is your sensory neuron. Then in between that sensory neuron and the motor neuron that will do something is this interneuron. So it is getting information, processing, and communicating to your motor neuron. The motor neuron is then sending this signal out away from that central nervous system to the muscles to do something. Okay. Let's look at the synapses. Synapses are the place where a neuron connects to another neuron or an effector. And there's two kinds of synapses, chemical and electrical. chemical synapses are much, much more common than the electrical synapses. So, of course, let's start with electrical synapses. They're not as common. You've got a presynaptic and postsynaptic neurons that are stuck together with a gap junction. And then the electrical signal goes from one because of that gap junction, goes right to the other. There is no delay in passing this electrical signal. It's very instant. The chemical synapses are much more common and they do have a delay. So we've got the presynaptic neurons. You've got this axon terminal that is going to produce a signal. Then you've got this postsynaptic neuron that will receive the signal. And this postsynaptic neuron is going to receive it at one of its dendrites. The little area that is in between those two neurons is called the synaptic cleft. So how do these two neurons communicate? Well, step one, neurotransmitter molecules are released from vesicles on the synaptic knob. Those neurotransmitters go into the synaptic cleft. The neurotransmitters continue to diffuse across the cleft. They bind to the postsynaptic receptors. Once they are binding to that receptor, then there'll be this initiation in that postsynaptic neuron. We're going to initiate this potential. And because we have to move neurotransmitters across this cleft, there is a delay. And that is just the time it takes for those events to happen, for there to be a release of the neurotransmitter, an uptake of that neurotransmitter, and then the nerve can act. So what are glial cells? We also call them neuroglia. These are not able to be excited. They're sort of the little support or helper cells that are in the central nervous system and the peripheral nervous system. They are small, and they do outnumber neurons, and they account for about half of the volume of the nervous system. They are able to do mitosis. They are there to protect, to nourish neurons. And they act as like a scaffolding for which nervous tissue develops. So they sort of guide neurons to where they need to go in development. And they're really important for the neural synapses. They're vital to that function. So with that, we have some types of glial cells. We've got astrocytes. They're shaped like stars. Use your imagination a little bit. But I like astro, like space, astronaut, and they're star shaped. They have processes that end in something that we call perivascular feet. They are the most abundant in the central nervous system, and they make what we call the blood-brain barrier. Basically, they wrap their little feet around the capillaries in the brain, so it controls what can go into the brain, which you can imagine is incredibly important because we don't want everything that is in our blood having access to get into our brain. We don't want virus or too much of an ion. We want that in the blood being taken care of. They also regulate tissue fluid composition, or what is around neurons. So they can really regulate potassium concentration. They're going to form support for other neurons. They assist in neuronal development, or like, organizing them in growth and development. And they will take up space of dying neurons. We have ependymal cells, which line cavities in the brain and spinal cord. It's part of what we call a choroid plexus, which is what makes our spinal fluid. You have microglia, which are really small cells that sort of wander around the central nervous system. Um, and if there's infection, they'll replicate. They're part of the immune system. They'll basically remove debris and any harmful substance. And then you've got oligodendrocytes, which are big cells that have very skinny fingers or projections. And those will wrap around axons of neurons, forming myelin sheath. So we've got, our neuron that we've been talking about with our dendrites, our cell body, our axon, but we've got these other accessory glial cells that are protecting and helping keep a structure, an order, a safety to our nervous tissue, our nerve cells. So again, we've got those astrocytes. You got to use your imagination a little bit that this looks like a star wrapping its feet around the capillaries. We've got the ependymal cells here that's there, that are lining the cavities of the brain and spinal cord. You've got microglia cells that are protecting and preventing infections, and then the oligodendrites making this insulation myelin sheath area. So let's look a little bit more at glial cells because those were our main ones, but let's put them into now our peripheral nervous system. We have what we call satellite cells. They are arranged around these neuronal cell bodies. They are there to insulate and regulate the exchange of nutrients and wastes. Then we've got what we call Schwann cells or neurolimocytes, which are long, flat cells that are going to wrap around the peripheral nervous system axons. And those are insulating them and allowing for a faster action potential propagation. So moving that electrical pulse, basically. So that's here where you've got, again, what we've been calling our axons. We've got our dendrite and cell body here. Here's our interneuron. Okay, so we've got satellite cells here that are insulating the cell bodies and regulating what nutrients come in and out. And then we've got the Schwann cells here wrapping around, insulating to allow for faster propagation of that signal. So, interesting clinical view. I don't always keep these in, but it's fascinating stuff, and I know a lot of you are wanting to do medical careers in some way. So let's, tumors on the central nervous system. What's happening there? So neoplasms are unregulated cell growth. That's a tumor. And these can occur in the central nervous system. So there's tumors that are in the brain. Okay, then we would call them primary brain tumors. And we can have things that originate in supporting tissues. Like that's where it's happening. So those tissues are doing mitosis. Specifically, one kind is like in the meninges, that protective outside part on the brain and the central nervous system, or in glial cells. So we'll actually have something called gliomas, which are glial cell tumors. They can be benign, but if they're malignant, then they metastasize. And obviously that would be bad. They could infect other tissues and other parts of that central nervous system. Okay, myelination. I've said myelin a whole bunch, but what is that? So myelination is just wrapping the axon with myelin. And that's going to be, again, for insulation and quickly sending that signal down the neuron. So there are several layers of, myelin is several layers of glial cells wrapped around. It has a high lipid content, so it has this sort of white, shiny appearance. It's going to insulate the axon, and in the peripheral nervous system, those are the neurolemmocytes, and it's the oligodendrocytes in the central nervous system. In the peripheral nervous system, you've got that neurolemmocyte encapsulating that neuron axon. It wraps it in layers, and we call it myelin sheath. The neurolemmocyte cytoplasm and nucleus are kind of pushed off to the side, and it forms what we call a neurolemma. The neurolemmocyte can myelate only 1 millimeter of axon. So we need a lot of these. Obviously, we need more than 1 millimeter. So we have several of them wrapped around. Any gaps that are in between one neurolemmocyte and the other in creating that myelin sheath are called neurofibril nodes or nodes of Ranvier. And here's what we've got. So this middle part, that's your axon, that's your nerve, and you need to wrap it up in those sorry, lost my word. Things are happening in my apartment. We're going to wrap that up in our neurolemmocyte here, and we're going to push that nucleus off to the side. It's no longer really useful and needed, and now we can wrap it with several of these, and we've got a myelin sheath that has been created. This little piece here, like that's an opening, that's going to be your node. Okay, here's a bigger picture if that is helpful. Again, remember dendrites, cell body, axon here, axon terminals. Here's your neurolemmocytes all around it, wrapping it up, creating that myelin sheath. Here's your neurolemma, which is that nucleus kind of pushed off to the side, and any little spaces in between, that's your neurofibril node or nodes of Ranvier. Okay, central nervous system. One oligodendrocyte can myelate 1 millimeter. And with that, we're not forming our neurolemma, right? That's not pushing it to the side. Instead, we still have those nodes between adjacent segments. But basically, the main difference is instead of pushing the nucleus to the side, it's not pushed to the side, it's still here, but we're able to wrap up about 1 millimeter at each spot. Okay. So there are unmyelinated axons. In the peripheral nervous system, the axon is going to sit in a depressed portion of that neurolemmocyte. So it sort of fits in here like, I don't know, if you're taking like a stick and putting it through some Play-Doh or something. So it's not totally wrapped and encased. It's just kind of laying in there. And here, so we're just laying in here. And then in the central nervous system, the unmyelinated axons just aren't associated with oligodendrocytes. That's what's happening. They just don't have an association. Okay. So what does this mean for disorders affecting that myelin? So a very commonly well-known disorder is multiple sclerosis. This is where there is a progressive demyelination in your central nervous system. It is an autoimmune disorder. Basically, those oligodendrocytes are being attacked by the immune system, and it's causing inflammatory events, which causes scarring, which causes loss of function. So you will lose the ability for those nerves to basically transmit the signal, which means you'll lose the ability to, control that nerve and control that body organ that it's affecting. And then there's Guillain-Barré syndrome, which is losing the myelin from the peripheral nerves due to inflammation. Muscle weakness starts in the far limbs and advances. And then the most function recovered with little medical intervention. Or multiple sclerosis, we don't see that. But both of these disorders are because of the myelin being affected. Okay, so here's where we'll get into that axon regeneration. We said before that most nerve and nerve tissue, like once it's damaged and dead, that's it. That being said, there are some axons and nervous tissue that we can have regeneration in. So after traumatic injuries, the peripheral nervous system axons, that's a big one, peripheral nervous system axons can regenerate if the neuron cell body is still attacked, there is enough neurolemma remaining. So not only is it just the peripheral nervous system, this regeneration can only happen if the cell body is intact, there is enough neurolemma remaining. Regeneration success is even more likely if the amount of damage is not extensive and the distance between sites of damage and the structures that it innervates is short. How does this regeneration happen? Step one, there has to be trauma, obviously. Step 2A, there is these axons that will seal off and swell. So that's close proximal to this cut in the nerve. The axon seals off and swells up. Farther away from this cut, the axon and the sheath will degenerate, and we call that Wallerian degeneration. But the neurolemma is surviving. Step 3, the neurolemma and the endoneurium are going to make what we call a regeneration tube. Axons regenerate guided by nerve growth factor. So that is going to be released from neurolemmocytes and stimulate or it guides this nerve growth. And then the axon is going to re-innervate the original effector or sensory receptor. So again, here is a picture image of what is happening. You've got your nerve, right? It is acting normal, right? Dendrite, cell body, axon. Okay, there is a traumatic event. Damage happens. We've severed this nerve. Okay. First thing is we have to, split it off close to that cut and it will swell up. Okay, that's, I guess, here to here. We will then make a regeneration tube in which we are able to then regenerate with our growth factors where we're making that axon grow, and then we are going to innervate that same place, that same effector or sensory, whatever it was, we innervate that same thing, and now we've regenerated this nerve. In our central nervous system, it is incredibly limited, difficult, not really common to see regeneration. You have the oligodendrocytes secrete growth inhibiting molecules, not growth factors. And there's a lot of axons in the central nervous system. So regrowth really gets obstructed from these scars, and it doesn't really happen. Okay. That being said, there can be some rare circumstances where we see it, but for our purposes, regeneration will really happen only for peripheral nervous system nerves. If this is something that interests you greatly, I highly encourage you to watch some of the videos on the D2L, to do your own research, because it is incredibly fascinating. But for our purposes, peripheral nervous system regeneration only. Okay, we are going to switch gears almost entirely to talk about pumps and channels, so we can finally get into how we're moving this through our signals through our nerves. So first, pumps. Pumps are membrane proteins that will work to maintain a concentration gradient by moving substances against the concentration gradient. So if there's a lot of calcium or iron or whatever it is outside of a cell, and we still want to get more outside, we need a pump. Pump will put it from inside the cell, outside the cell, against a gradient. This is going to need ATP. Neurons have sodium-potassium pumps and calcium pumps in their membranes. So the three major ions, sodium, potassium, and then calcium. Channels. Channels are different. That is little pores in which ions are allowed to move down their concentration gradient. Does not require energy. There are leak channels that are always open to allow continuous diffusion. There are chemically gated channels that are normally closed, but will open when a neurotransmitter binds to it. And there are voltage gated channels, which are normally closed, but open when the membrane charge changes. Let's look at the voltage gated sodium channels that we find in our neurons. So one phase that we can find is the resting state. That means that the activation gate is closed, the inactive gate is open, which means we are not letting sodium into the neuron. The second stage we can find is what we call an activation stage. The activation gate is opened because we've changed the voltage on the membrane. The inactivation gate is also open. That means that sodium can move through these channels. Then there's the inactivation state. The activation gate is open. The inactivation gate is closed. Sodium cannot enter anymore. And this is a very short amount of time because the channel will quickly reset to the resting state. So here's what we're looking at. You've got your cytosol at this bottom part. That's the inside of your cell. And then you've got the outside up here. Normal conditions, we're seeing that there is a lot of sodium outside of the cell and a little bit inside. When we're at our resting state, this is closed. Sodium can't get in. When it's activated, this gate opens. Sodium can go in. And we see we went from a positive charge to a negative charge. That's let sodium come in, right? It's voltage gated. Then we've got an inactivation state. This is very brief, where we still have this gate open, but we are not letting more sodium in. And it'll quickly go back to this stage. Okay. Because they have these two gates, they can be in all three of these states at a slightly different rate, which is how we propagate or move that signal. Okay, to change gears just slightly again, you have modality gated channels. Normally they're closed, but open in response to a stimulus that is not chemical or voltage change. These are in the membranes of sensory neurons, and they respond to changes in the environment. A great example is in your skin. You have some modality gated channels that open in response to mechanical stress or pressure. So, where are these pumps and channels? And we said, we can have them be at the three stages, slightly askew from each other to move this potential or this signal down. So, like, where are we finding these? Throughout the entire plasma membrane of a neuron, you will find leak channels, specifically potassium leak channels. Potassium leak channel, always open. Things can move down their concentration gradient, diffusion of potassium. You will also find the sodium potassium pumps, the thing that has a little gate that will open and close based on that electric signal. The job of those two types of pumps is to maintain a resting membrane potential, to allow the outside to have a positive charge, the inside to have this negative charge. In the plasma membrane, you'll have functional segments in the neuron. You've got a receptive segment. This receptive segment is in the dendrite and cell body. In that receptive segment in the dendrite and cell body, you have chemically gated channels. Chemically gated chlorine channels, to be specific. Chlorine, okay? Then you have that initiation segment. That's that part where the axon meets the cell body. That is voltage gated sodium channels and voltage gated potassium channels. You've got a conductive segment. That's the rest of the axon and wherever it branches off. That is going to be your voltage gated sodium and voltage gated calcium channels, much like the initiation segment. And then you've got a transmissive segment that's at your synaptic knobs, where you've got voltage gated calcium and calcium pumps. So I know you're thinking, why is this important? Well, we need to know what ions are outside or inside the cell. Do we need energy to move the ion, or does it just leak through? And that will help us to determine, sort of how we move this signal down. So knowing what channel, is it leak or is it gated, is important. Okay, so here's what we're seeing again. Here's your cell body and your dendrites. We've got chemically gated channels here for our sodium and our chlorine. If we go to this initiation part, it's our sodium and potassium gated channels. As we go down this axon, it's still that sodium and potassium channels. And then throughout the whole neuron, whether it is the dendrites, the cell body, or the axon. You've got leak channels for potassium and sodium, and potassium-sodium pump. When you get down to your synaptic knob, you are also going to have voltage-gated calcium channels and calcium pumps. So what we're moving when we move these ions is really an electrical signal. Okay, electrical energy is just the movement of charged particles. And neuron activity depends on that electric current, right? We're moving ions electrically or charged particles, which makes it electrical energy. When we talk about electrical energy, we have to talk about voltage. Voltage is the potential energy. That is the amount of difference in electrical charge between two places, whether that is in or out of the cell, right? That's our two places. And we measure this in volts, but for our purposes, we measure it in millivolts. Then there is current. Current is the movement of the particles across that barrier. So we're moving it along. And that, movement across that barrier, that membrane, is harnessed to do work. Resistance is the opposition to movement. So it's the membrane, it's the barrier. And an increase in that resistance lowers the current. With that, we have what we call Ohm's law. Current is equal to voltage divided by resistance. So the current is the voltage over the resistance. As the current increases with larger voltage, the resistance decreases. If we're looking at neurons, charged particles that we're looking at are ions, and current is generated when the ions diffuse through the channels. Voltage exists across the membrane because of unequal distribution of ions. The membrane is offering resistance to that ion flow, and this resistance changes because of the gated channels. When they are closed and not allowing things in, that is more resistance. When they are open and letting things in, that is less resistance. When our neurons are at rest, the ions are unevenly distributed across the plasma membrane. There is more potassium in the cytosol than in the interstitial fluid, so more potassium inside. There is more sodium, chlorine, and calcium outside of the cell. When we look at that, we can see then we're going to have a positive charge outside, a negative charge inside. How do I get that? Well, we've got +1 and +2, so that's +3, -1, +2, +1. So we have a difference in charge, okay? The gated channels are closed. Okay, gated channels are closed. There's electrical charge difference. The cytosol is more negative. Did I just say this backwards? I may have said it backwards. So go back, rewind. Don't listen to me. There's higher concentrations of potassium inside the cytosol compared to outside the cell. Higher concentrations of sodium, chlorine, and calcium outside the cell than inside. The cytosol is negative. Negative. Don't listen to if it was earlier I said positive. The cytosol inside the cell is negative. Negative. When we look at that, that means our resting membrane potential is negative 70 millivolts. How did we get negative 70? Don't worry. Someone else measured it with a millivolt reader. That's all you need to know. Negative 70. There you go. Right there. Don't worry. You don't have to do that. Just remember negative 70. So here's what that looks like in a picture form. We've got our potassium inside. We've got calcium and sodium and chlorine outside. That means we have a negative potential, right? Our channels are closed. Okay. That potassium diffusion is the most important factor in our resting membrane potential. If potassium diffuses out of the cell due to its concentration gradient, diffusion out is limited by the electrical gradient, the pull of the negative resting membrane potential, right? So we have this negative charge. Potassium wants to go out, but it can't. It's being held in place. If potassium were the only ion that leaked, then the resting membrane potential would be where the potassium concentration and electrical gradients equal each other. Okay, but that's not happening. Since there are a few sodium leak channels and sodium also influences this, sodium moves in because of its concentration gradient. This small leakage is what gives us our negative 70 millivolts. That's what we need, negative 70 millivolts. That's our starting resting potential. What does that sodium potassium pump do? Well, it's going to push 3 positive charges out. Okay? 3 potassiums out, and it's only pushing in 2 sodiums. So every time we're doing that, pushing 3 positives out and 2 in, we are contributing to this resting membrane potential about negative 3 millivolts. Again, that's just to maintain that negative 70 total. Most importantly, this sodium potassium pump is just keeping a concentration gradient. It is keeping more potassium in, more sodium out. Okay, that's what we need. We need this sodium and chlorine outside, potassium inside. Potassium wants to leave, but it has a hard time doing that. Okay, that's what we've got. So let's have a little overview of these segments again. You've got your receptive area, this initiation area, this conductive segment, and then the transmissive segment. Okay. What we were just talking about really is an overall concept here, because we've got this resting potential. But now we're going to go ahead and go into the receptive segment. Graded potentials are small, short changes in the resting membrane potential. Graded potentials are small, short changes in the resting membrane potential. And it has to have these following characteristics. 1, they are established in the receptive segment by opening chemically gated ion channels. The current changes are local and short-lived. They vary in degree of change and direction of change of the resting membrane potential, which means it can be a large fluctuation or a small fluctuation. And they cause depolarization and then hyperpolarization. What does this look like? Graded potential in the postsynaptic neuron is called postsynaptic potentials. The postsynaptic potential results in the depolarization, and we call that the excitatory postsynaptic potential, or EPSP. Right after that happens, the postsynaptic potential resulting in hyperpolarization going right back up, is called inhibitory postsynaptic potential, or IPSP. Numerous postsynaptic potentials can be generated because postsynaptic neurons bind to many neurotransmitter molecules at the same time. So basically, you're at the part receiving the signal, and we can have lots of these potentials happening all at the same time. Okay. So here's that. You've got this other axon terminal releasing the neurotransmitter. When it's binding here at all of these different spots, that is that excitatory postsynaptic potential. And that is depolarization caused because sodium is allowed to enter. We open the sodium channels. The inhibitory response, or inhibitory potentials, are hyperpolarizations caused by potassium exit and chlorine going in. So if you looked at this little graph, when you had the EPSP, there was a little hump. Now we've got this little divot. And this graphic is just showing you that, here we have the excitatory and the inhibitory all happening at the same time. So we can have all of this going on at once. So summation is just taking all of those EPSPs and all of those IPSPs and adding them together. So all of those little humps that go up to the little dips that go low, adding it all together. And we do that at that axon hillock, the part where we're getting the cell body to the axons, that little triangle area. Voltage changes from the dendrites and soma are added. The sum may or may not reach the threshold to initiate action. The threshold is the minimum voltage required to make that change, to open those channels, to propagate that signal. Typically, we say the threshold is negative 55 millivolts. And that is a number, sure, you can remember, but typically, generally, it's negative 55. As a whole, we're going to need multiple EPSPs added together. If the threshold is reached, then the voltage-gated channels open and that action potential is generated. We propagate that signal down. We can do this summation over space and over time. Spatial summation is multiple locations on a cell's receptors are getting neurotransmitters simultaneously. Temporal summation is a single presynaptic neuron repeatedly is releasing that transmitter and is making the EPSPs. So here's what it looks like spatially. Lots of EPSPs and IPSPs are happening at this one cell body. Temporally, it is still looking like this in picture, but you've got one space, whether it's, right here, that's constantly sending this. It's almost like shaking a person awake, being like, hey, get up. That's what's happening. One specific spot is like, hey, this is happening. And that is going to excite a response if we reach the threshold. With this, there is a all or none law. If the threshold is reached, action potential generated and goes down the axon, and we don't lose intensity. Okay, that's the all. If the threshold is not reached, then there's no action potential. That's the none. So we either fire the gun or we do not. And then the axon shows the same intensity of response to values greater than the threshold. So the example your book gives, if you have sufficient pressure on a trigger, you fire a gun. If you don't fire the gun. If you squeeze the gun as hard as you possibly can, it still fires the same as if you just reached the threshold. There's no difference. All right, so now we've got the receptive part. We've got the initiation part. Let's get the conductive part of this axon. So the axolemma is what conducts the potential. Action potential involves depolarizing and then repolarizing. Depolarizing is the gain of a positive charge as sodium goes into the sodium channels. Repolarizing is returning to negative as potassium leaves the potassium channels. The action potential is propagated down the axon to the synaptic knob. So voltage-gated ion channels open sequentially down that axolemma. That's like what we very first talked about with the channels, is that you can have all three states happening one right after the other in succession. Okay. That propagation is called a nerve impulse or a nerve signal. And basically what's happening, you've reached the potential that this is happening. Action potential happens. How do you get depolarization? Step one, when we're at resting membrane potential, voltage-gated channels are closed. Then sodium is going to enter from an adjacent region, opening the channels. The sodium enters the axon, causing the membrane to then become a positive charge. The sodium channels will close and become inactive temporarily. And then we just move on to the next channel and do the same thing. So we are at our resting potential. We've reached the threshold. Now we're building and pushing this signal. Okay, step 5, depolarization happens more slowly because we open the calcium or the potassium channels. Potassium diffuses out, and we are then going back to a more negative potential. Those potassium channels will stay open. So potassium exits, and then we go back past hyperpolarizing, past our negative 70 resting potential. Eventually, we will close those potassium channels and we go back to our negative 70 millivolts. And that repeats as you move down towards the synaptic knobs. So here's what this would look like graphed out. Resting potential. Now you've got your graded potentials where you're getting those ISPS and ESPSs. We reach the thresholds. All or nothing depolarization. So rapid. Repolarizing. Potassium channels stay open. Hyperpolarizing. Back to normal. There is what we call a refractory period. That's the time after that action potential has started when it's impossible or incredibly difficult to fire another action potential. There is the absolute refractory period, which is like 1 millisecond. No stimulus can initiate another action potential. Those sodium gates are open and then they're inactivated. This is ensuring that propagation goes all the way to the synaptic knob and it doesn't reverse or stop. Then there is this relative refractory period right after the absolute refractory period, where we could get another action potential, but the stimulus would have to be greater now. And then this is happening because the channels have reset. Some potassium channels are still open. The cell is still hyperpolarized. So you would need a higher level of those ESPSs, a higher stimuli to get another firing. Okay. And that's just showing you here. You're below the resting potential. So you would need more stimulus to get to that threshold. In that conductive segment, there is continuous conduction and saltatory conduction. So continuous conduction is happening on unmyelinated axons. The charge opens voltage gate channels that allows charge to enter. It spreads to adjacent region and opens more sequentially. That's what you normally think of. That saltatory conduction is on myelinated axons. Action potential only occurs at those nodes, which is where the axon's voltage gated channels are concentrated. Then sodium enters the node and it starts rapid positive current down the inside of the myelinated area. The current becomes weaker with distance, but it's strong enough to open the channels at the next node. So at the next node, more ions are coming in, going down that myelinated segment to the next node, open, and so on and so on. The full action potential occurs at the node and the process repeats, the impulse seemingly jumping from the node to node. This is actually faster than continuous conduction, and myelinated cells use less ATP. So while it doesn't feel like it is faster or easier because of how it's going in at the node and spreading down and in at the node and spreading down, it is faster for your body, uses less ATP. So this is just showing that. Here's your nodes where the sodium is rushing in, going down, opening up that channel there, rushing in, going down, opening up there, rushing in, going down, as opposed to here where there's no myelin and all of these different channels have to open. That's that difference in the ATP. It's almost like there's less that you have to do when you're myelinated. Okay, our transmissive segment, that is the synaptic knobs. So you've gotten your action potential arriving at this area, and that action potential will open the calcium channels. And calcium will diffuse into the synaptic knob, and those calcium pumps already made a gradient. And the calcium will bind to proteins associated with vesicles and trigger exocytosis. Those vesicles fuse with the membrane at the synaptic knob, and neurotransmitters get released. Subsequently, neurotransmitters bind to the postsynaptic receptors, and that may trigger this whole process in that following neuron. Neurons can synthesize more than one neurotransmitter, but only one type is released at a time. And depending on the frequency of action potentials, the reach of the synaptic knob is sort of how it's uptaking those neurotransmitters. So here is what's happening. We've got that action potential that has reached the knob here, and that has caused these calcium ion channels to open. They have rushed in to attach to these vesicles that now are like, okay, I got to move, and I release my neurotransmitters, which are then taken up by the next neuron. We have to look at graded potentials and action potentials side by side here. Graded potentials occur in the receptive region because the ions flow through those chemically gated channels. They can be positive or negative. They have a larger potential change to a stronger stimulus, which means you can still fire if you increase the stimulus. And they are local and travel a short distance. Action potential, that's over the entire conductive region as ions flow through. It'll have the depolarization and repolarization. That's our all or nothing, and it's going to go to the synaptic knob. How fast is this really going? Well, it's going to depend on the axon and its diameter and its myelination. Thicker fibers conduct faster than thin ones. Thick axons have less resistance to current flow. Myelinated fibers conduct much faster than unmyelinated ones because going from node to node is very fast. You also have something called nerve fiber groups. Nerve fiber, that's the axon and its sheath. You can have group A, which is about 150 meters a second. These are the large diameter myelinated fibers. It's a lot of your somatic sensory neurons, all of the somatic motor neurons. And then there is group B, which is 15 meters per second. And then also group C, which is 1 meter per second, which is small and unmyelinated. It's some visceral neurons, some somatic sensory neurons from the skin. Do you need to know exactly where those are? No. If you want to know exactly where those are, then I encourage more research, but just knowing about group A and about group B and C is enough. Impulse frequency, or how many action potentials per second, does vary. So although the impulse amplitude is constant, right? We're going from negative 70 and then below that and back to it. The frequency varies with stimulus strength. For example, bright lights cause faster firing frequency on the optic nerves than the dim lights. When motor nerves fire at faster frequencies, it can cause more muscle tension. And for neurons that can use different neurotransmitters, the firing frequency can influence the kind of neurotransmitter that gets released. So neurotransmitters, that's kind of our last big thing here. What are those? Neurotransmitters are synthesized by neurons and stored in vesicles in the synaptic knobs. They are released from vesicles when action potential trigger calcium into the synaptic knob. They bind to receptors in target cells. And they trigger some sort of physiological response in the target cells. There's 100 or so different ones that have been named and classified. We are not going to do all 100, but there are some common and important ones we will look at. So 4 main classes: acetylcholine, and that structure differs a lot from other neurotransmitters. Biogenic amines or monoamines, that's an amino acid that's been modified, and within that you've got catecholamines, dopamine, or indolamines, serotonin. And if you know dopamine and serotonin, those are like happy feeling, loving feeling. And there are medications if people are suffering from depression or anxiety that really help the brain like uptake serotonin specifically. But they are in the biogenic amines. There are amino acids that alone act as transmitters, like glutamine, glycine, and GABA or GABA. And then there are neuropeptides, which are chains of amino acids, about 2 to 40 amino acids, including endorphins and something called substance P. Neurotransmitters are classified by their function. Classification by effect on the membrane potential is one way. So, excitatory transmitters cause EPSPs, inhibitory cause IPSPs. Some transmitters can excite some targets and inhibit others, just depending on the receptors. So you don't have to get into that too much, but just know some can do both things. They can also be classified by action. There are direct transmitters that bind to receptors. That receptor is a chemically gated channel. So you've got immediate transferring, immediate postsynaptic potential, right? Moving that right away. Some are indirect transmitters that they bind to receptors that involve a G protein and secondary messengers. So it's not an immediate or as immediate because of that. But there can be more diverse effects possible, right? So if you're stimulating a chain of reactions, there's more pathways where different actions can arise as opposed to the direct transmitter. Okay. Again, do you need to know all 100 of those different transmitters and where they fall on these two charts? No. Should you know that we classify based on chemical structure, classified based on function? Yes. Yes. Is it cool to know a few like serotonin and dopamine and GABA? Yes. Do you need all 100? No. I'm not sure why this question comes up, but it does often. I've never made you memorize 100 of something. I would not start now, I promise. Okay, let's look at acetylcholine. That's the most common, best characterized neurotransmitter. It is used in the peripheral nervous system to stimulate skeletal muscles, and it's used in the central nervous system to increase arousal. It is made from acetate and choline, which are stored in synaptic vesicles. Action potential triggers it to be released into that postsynaptic cleft. Some acetylcholine attaches briefly to postsynaptic receptors. Acetylcholine is cleared from the cleft by being broken down to acetate and choline by an enzyme called acetylcholinesterase. You don't really need to know that, but it's broken down by an enzyme. Acetate and choline are then taken back up by the presynaptic cell and are recycled. There is the effect on target cells depends on the receptor present. There's two receptors there, nicotinic and muscarinic receptors. Okay, removal of neurotransmitters from the synaptic cleft can happen in different ways. In what we just talked about with acetylcholine, there's an enzyme that breaks it down. There are other ways. So the enzyme might degrade a neurotransmitter, step one. You could also have presynaptic transporters that import neurotransmitters. We call that reuptake. And so, again, going into like anxiety, depression medications, SSRIs, selective serotonin reuptake inhibitor. So, we're not allowing serotonin to be reuptaken with those medications. But basically, they're importing it back in. Some new neurotransmitters diffuse away and the glia absorb it. So what I just said, some drugs have effect on that. Selective serotonin reuptake inhibitors treat depression. So serotonin, one thing with depression and anxiety is that your receptors for serotonin aren't working as efficiently. So serotonin that should be causing your neurons to have that next firing, being locked in by their receptors are not. And so that serotonin would then be reuptaken into the presynaptic vesicles, and you don't want that. So you block that with SSRI. You could also have something like galantamine hydrobromide, which is an acetylcholinesterase inhibitor. So it blocks the enzyme so that acetylcholine isn't broken down, and that's used to treat Alzheimer's. So here's what we're looking at there in particular. You've got acetylcholine that has been released from the presynaptic cleft, and it's in this cleft, and it's wanting to go here. So you could have that enzyme to break it back down into choline and acetate. Well, if we block that enzyme, that will not happen. And then we can keep taking in acetylcholine to this post-synaptic cell, which is again a treatment for Alzheimer's. Neuromodulation is a little subsect of this. Neuromodulators are chemicals that alter responses of neurons. And so you've got facilitation, that's a modulation that creates a greater response in the postsynaptic neuron. So it increases the amount of transmitter in the cleft or the number of receptors. Then there's inhibition, which causes a weaker response. You decrease the neurotransmitters in the cleft or the number of receptors. Examples, nitric oxide, that can be used as a neurotransmitter modulator. It's very short-lived. It's a nonpolar gas. It's laughing gas. And it is made and released by postsynaptic neurons in the brain, where it's believed to strengthen memory by affecting presynaptic cells. And it affects the peripheral nervous system, including blood vessel dilation. And then you've got endocannabinoids, which influence same receptors that marijuana does. And it is a small nonpolar molecule made and released by postsynaptic neurons and have effect on the amount of neurotransmitters released. And that can influence memory and appetite. We also have something called neuronal circuits or neuronal pools. Those are groups of neurons arranged in specific patterns. There's really 4 kinds of circuits: converging, diverging, reverberating, parallel after discharge. And these pools can be localized or distributed in several regions of the central nervous system. A convergent circuit means that the input converges at a single postsynaptic cleft. So there's multiple sensory inputs, like, for example, neurons in the salivary nucleus, sight, sound, smell of cooking, all lead to you salivating. Okay, so you've got nerves in your eyes and for your nose and touch and sound and smell, and that all converges to make you salivate when you're cooking. Divergent, that's spreading the information out to multiple postsynaptic neurons. So when you're controlling something like walking, that's sending several different signals to different muscles for posture, balance, motion, actually moving your limbs. So that's divergent circuits. Reverberating, it's a little more complicated, I guess, in that it uses feedback to produce repeated cyclical activity. So once we start this, it stays active until there is an inhibitory stimulus or fatigue. An example is the circuit that keeps us breathing regularly when we sleep. And then parallel after discharge, that's the input is transmitted simultaneously along several paths. But since the paths vary in number of synapses, the signals arrive at different times. And really, this is, I don't want to say more rare, but a more rare circumstance, but it's believed to be involved in higher order thinking. So it's something that we find in humans that we don't necessarily find in animals, which is something that makes us unique. Okay, cool. So we're going to end it there. Again, if you have questions, please feel free to reach out to me. I'm more than happy to help. Again, there are videos and worksheets and Quizlets and all kinds of things on the D2L for you as well to dive into this. But otherwise, thank you for watching, and I hope you have a wonderful rest of your day.